Hello and welcome to Long COVID Foundation podcast. Are you seeking answers and solutions for the lingering effects of long COVID? Well, you've come to the right place. This is your go-to channel for educational resources tailored to help you understand and cope with the challenges of long COVID. Whether you have been following us from the beginning or just joining our ever-growing community, we extend a warm invitation to hit subscribe button and become a part of our ever-curious and supportive audience. So far, we learned a lot about the virus and, of course, long COVID. We covered topics related to genetics, gut health, microclotting, gut-brain axis, cardiology, neurological issues, and many, many more. And I encourage you to watch previous podcasts. While doctors are still trying to understand long COVID, we present the most up-to-date scientific findings on SARS-CoV-2 pathogenesis and, what's more importantly, ways for your recovery. And great news, we are launching nutritional sessions as well as simple and healthy recipes for you to get nutrient-dense food on your plate. Because we know as we are what we eat. After today's webinar, you will understand the connection between severe long COVID cases and dysbiosis, which is seen among many patients. So if you want to learn more, subscribe to this channel and follow our work. Thank you. And before I will present our guest today, I want to read a foreword nicely describing our current situation as researchers. In January 1610, Galileo Galilei observed 4 celestial bodies similar to stars near the planet Jupiter, using a telescope of his manufacture, not even certified yet. 2 months night work, unpaid and uninsured, led to the discovery of Medici stars, the stars that revolve around Jupiter. Thanks to scientific development, we know that what the Pizan called stars are actually Galilean moons, and their exact number is 79, not 4. Fortunately, Galileo didn't receive a Nobel Prize or even a doctorate. Thus, he managed to avoid spending significant amount of his time on meetings, webinars, articles, publishing, and double, if not triple, judging. Galileo wrote a simple book, illustrated by hand, with no bibliography, to share his observations with everyone. Yet every self-respecting scientist in the world knows what the Galilean method is. Well, dear listeners, subscribers, and just followers, whether you're a qualified academic researcher or just an ordinary human being, you should know what Carlo will discuss today and what he proposes is the result of intuitive actions, simple tests, and observations. I have mentioned this astronomer because I'm trying to inspire you to improve and enhance the new approach to the study of SARS-CoV-2, because it is important for our patient future treatments and successful recovery. With all my respect for this historical figure, I'm not trying to compare Carlo, myself, or anyone from our research team in any way, but we want you to understand a novel way of thinking about SARS-CoV-2. Carlo, we all are waiting for your presentation today and discussion about your scientific findings and this groundbreaking research that you have demonstrated to us all. Welcome. Carlo, welcome to our channel, and I will give you a possibility to share your research in detail, starting back from 2020, where you started all your discoveries. And I'm really proud to be part of your research team and collaborate on small bits and pieces together with you. And please talk us through your findings in a way that our community, our long COVID sufferers understand, and please explain to us the main mechanism that you have discovered and what role it plays in navigating future treatments. And let's discuss today the paper which recently, just a couple of days back, came out in Journal of Medical Virology with the impact factor of 20.9, which is a significant impact journal where you and your team published this groundbreaking research. Really well done for your work and your inspiration and your hard work that you bring with no extra funding, with no public contributions to the work that you do. So please welcome Carlo to our channel.
Thank you. Thank you so much. I appreciate very a lot your presentation. Thank you. For me, it's enjoying stay here with you and all the friends of your channel. And we have make many road along these 4 years, and we have fight against other research and other opinion. We have an idea, and this idea was not of mine, but I believe in God, and I believe that we have many help by our Creator. So thank you for my invitation, and I will share my presentation, and we talk in summary about, or we discuss in summary about 4 years of research that are concluded with the paper, just like you say, that was published 2 days ago in Journal Medical Virology. It is a paper that came from a long research before starting with laboratory data on bacterial culture and SARS-CoV-2. We have found many toxin-like peptides in blood and of patients with COVID-19 and derived from gut microbiome infected, and we also have discovered the combination of two drugs that in this case are antibiotics that from one side can stop the toxin production and at the same time can make a low viral replication and control the infection of the virus SARS-CoV-2 against the bacteria of gut microbiome. So we can start if you want.
Thank you. Yes.
My presentation is focused on the realm of bacteria and the gut microbiome and also microbiome of the human being. So everything you need to know from the perspective of microbiome. I want that everyone fix in his or her mind this picture of bacteria infected by viruses. Okay, and there is also a spectrum, or we say precisely a peptide mapping of an experiment that later I will show you to everyone. And in this experiment, we make a very nice test. Bacteria in general. Bacteria have enabled life on the planet. Bacteria help us to live on planet. Bacteria will continue to exist after us. Bacteria are the true witness of creation, because they are present by very, very long time. And to think doing science without first checking bacteria, I believe is pure ignorance. Some question I have at the beginning of this pandemic. What is SARS-CoV-2? Is SARS-CoV-2, everyone know now that is a RNA virus. What are the RNA virus? Are virus that binding to the epithelial cell, to the human cell, or virus that binding to the bacteria? So this is a lack in history of medicine and biology. A lack that it is very far away, long, many years long away, and no one have checked before or tested if viruses RNA can infect binding the bacteria or gut microbiome. So no one ever think it that bacteria can be the carriers of this virus. Only a few doctors in the world in the past years have studied this aspect. Uh, One is Luc Montagnier, the other one is Professor Zayak or Dedier, but only few doctors have studied or tried to study this aspect of the binding that could be between a virus to RNA and a bacteria. But when a bacteria was infected by a virus, we can classify the infection as also as a toxin infection because the gut microbiome we have show it in our paper that can produce toxin like peptide that we have found in the blood of COVID-19 patients. So there is a picture, a clinic picture, with connection between this toxin-like peptide produced by gut microbiome during the infection and the clinical aspect of COVID-19 patients. At this point, some question every one of us have made. What is the best solution? Antiviral, vaccine, vax, or what kind of vax? Is right to make an injection vax, or is better to give resistance to bacteria? What kind of antibiotics if the virus replicating in bacteria, and in which time is important to give the antibiotics? So this is all the question that we have made during this past 4 years. I decided that everyone who are listening this presentation fix in his or her mind this image of bacteria infected by viruses, or a bacteria lytic by a viruses. And all the all our study concluded with this recent and paper that we have published in Journal Medical Virology. So some doctors ask me why we have made a retrospective cohort study and not a prospective cohort study. This is the reason, a simple very reason. The Helsinki protocols of a trials don't admit that we make in condition of danger the patients when we well know the diagnosis about a illness. So we have first of all classified SARS-CoV-2 as a toxicological anti-infection illness for the production of toxin, and made we wouldn't make a cohort prospective study because if we don't give some products or drugs to the patients, we create the condition that this patient could be in danger. So we have made For this reason, I believe that there is only the possibility to do a retrospective cohort study. This is the very important ethical medical reason. So, what we have learned in the lockdown is what it means to lose a job or friend. We have a very horrible experience. 3 years later, SARS-CoV is still here, but someone have forget what is to do the real science. We have a listen. Science will win, or we are running at the speed of science. So this is the typical words that everyone have listened in these past 3 years. But who is the science? Why the science proclaimed? Who is self-proclaimed science? What does it look like? And so you have been studying the coronavirus family for over 50 years, including the spike protein, and no one have done check on prokaryotic cell before. This is the human science, not the real science. This is a human science that can make some problem. We have questioned this aspect of the science in the paper of gut microbes of Taylor, and we have claimed the importance of gut microbiome in the pathogenesis and transmission of SARS-CoV-2. We have claimed in our paper that there is a mistake, and this mistake is about don't check bacteria or gut microbiome. In one of our paper, increase of the first paper, our paper, increase of SARS-CoV-2 RNA load in fecal symptom, we have showed that SARS-CoV-2 can increase in bacterial culture, and we also show the mutation, the many mutation that bacteria can produce of SARS-CoV-2. And the other paper, we have talking about toxin-like peptides that are product by gut microbiome infected by SARS-CoV-2. And in the third paper, analysis of bacteriophage behavior of a human RNA virus, we have made a very simple experiment of nitrogen isotope, and we have shown how investigating if a virus bind first on bacteria. This is some images of our papers, and all the description of these images are in our papers. But what is important in these images is to understand that SARS-CoV-2 and RNA virus bind first of all on the bacteria of gut microbiome, and these bacteria produce mutation and produce toxicological events. This is the experiment that I spoke before, and I am speaking about. This is the experiment of nitrogen isotope. It is a simple experiment that show how to investigate a virus, and when we added the nitrogen isotope 15 in the culture, we can found this nitrogen in the proteins of SARS-CoV-2. And we have made another experiment with the poliovirus, because also poliovirus is RNA virus. And we wanted to check if also other viruses have the same behavior of SARS-CoV-2. And we have checked. So also poliovirus, we have make a paper on journal mass spectrometry about this experiment. Also poliovirus replicated and is still present in poliomyelitic patients, in the bacteria of poliomyelitic patients, in the gut microbiome. I want to remember to everyone that we have resolved the problem of poliovirus through the vaccination of Dr. Sabin, the oral vaccination of a good or friendly or no very effective variant or mutation of poliovirus. This is the explanation of the experiment of the poliovirus with the nitrogen isotope and in bacterial culture. And this is a table of Dr. Sabin. In these tables, Dr. Sabin claimed the evidence that the injection vaccine of SARC, the person who received the injection vaccine of SARC, who believe that with the antibody level they have the immunity, continue to produce virus and have the possibility to contract the virus of poliovirus. But who was naturally infected or who received his vaccine, oral vaccine, don't have or don't replicate more the virus of poliovirus. Sabine changed the endpoint of a vaccination that no one now want to remember. He changed the point from not the antibody level, but from the point of view of the mucosal immunity, surface immunity. So he claimed that no one replicated, and he changed the endpoint in the observation of the viral charge. So, in conclusion, never forget to check on bacteria for every other virus. It should be the first real postulated, and it should be applied to the first to cock postulated. In our paper, we have shown how the antibiotic that you see in this table can decrease the viral load on bacterial culture. And there are some other antibiotics that decrease the toxicological aspect. We have made a cohort retrospective study with the combination of an antibiotics that decrease the viral RNA load on gut microbiome and an antibiotics that decrease the toxicological aspect. The best one that we have obtained in our lab data and published is the amoxicillin with clavulanic acid and rifaximin from the other hand for the toxicological aspect. And this is the model that we have found or discovered. The model that SARS-CoV-2 infect bacteria and produce the toxic-like peptide. In other paper, we have observed also the effect of spike protein with the toxin-like peptide found in COVID-19 patients on the human 3D neuronal glia model. And we have observed that there is activation of the combination of these two aspects, spike protein and toxin-like peptides, can increase the activation of some genes. So, we are in the conclusion. This is our cohort study of about 200 persons. Some people are vaccinated against COVID-19 and others no. We have observed all the wave of this pandemic, the pre-Delta, Delta, and Omicron. And what we have observed with the combination of the two antibiotics that are for a early use in the first 3 days, and that the first endpoint is the blood saturation doesn't go down, but stay at a good height level in each group. The second endpoint that we have observed is that there is no difference between the two groups. And the duration of illness is the same for the two groups in about 7 days. So I know that many would criticize this paper, okay? But this is a paper with an history, and this is a paper with a lab data that no one would to do. We have checked in the lab first the binding of SARS-CoV-2 to bacteria and the binding of SARS-CoV and the production of toxic-like peptides. In this paper, we have observed also that the long COVID patients that we have treated early have the resolution of their problems with the use of rifaximin and two probiotics. I know that this is a very difficult position about our group, but what we claimed in this paper is that the first solution is to investigate the bacteria, and if we want to do a real good vaccination, it's better to re-study Dr. Sabin and poliovirus vaccination of Dr. Sabin, because we have understand with our experiment that also poliovirus bind to the bacteria. So it is very important to underline many and many times that the vaccination would be due after a good diagnosis, after to have observed if a virus can bind or not to the bacteria. Thank you to everyone.
Thank you, Carlo. Very good presentation, but I want to dig a little bit deeper for people to understand what is a real problem with bacteriophage behavior. So with that in mind, I wanted to share a very good image from the paper that Carlo just showed, and talk a little bit about the process of bacteria being infected by the virus, how it does it, how virus gets into the bacteria, and the importance of toxins which are released as a result of this process, how they destroy the tight junction and the risk of it getting into the bloodstream. Because this is the mechanism which drives all the symptoms that people have after COVID infection. This is the mechanism which never stops. And we need to do something about it. So can you go through this picture and signify the importance of this process? Because this is a real problem. This is why we discussed antibiotics. This is why we need to put on a scale what is best to kill the bacteria infected by SARS-CoV-2 early, or leave it to replicate and reproduce and get into the bloodstream through disrupted tight junction and epithelial cells on the gut lining. So this is very important picture.
Yes,
and
you are right.
And people need to understand this process. This is why we need antibiotics. This is why we discussed the importance of antibiotics in early stages, because we want to prevent this particular mechanism. And these toxins are very toxic. They get into the brain and they cause cognitive function issues in people after SARS-CoV-2. And we know that there is a clear link with those toxins related to thrombosis people are experiencing. So this image is something that I invite everyone to get familiar and understand in detail. So Carlo, I'll let you discuss it a little bit deeper.
Yes, okay. So how we have SARS-CoV-2 infect bacteria, and this was a problem, okay? But the bacteria are also intelligent because they, in the time, can produce resistance to SARS-CoV-2. But during the infection, there is as we have shown in the papers, there is a production of toxin-like peptides, and that we have stated that these toxin-like peptides are very similar to peptides that binding to the nervous system autonomous, to the receptor, nicotinic and muscarinic receptor, and also in the bloodstream and there are some toxins that are very similar to those one that create more coagulation of the blood. So in the acute phases, even if someone or some state or some institute say don't give antibiotics, we can understand this preoccupation about the point of view of antibiotic resistance. But we must also be honest and say to everyone that every of us sometimes assume antibiotics for other reason in our life. And in the major of this fact of this pandemic, it is stupid to don't use antibiotics if the right combination, because we have the good, we have see the good combination in the lab research. The good combination can stop the mechanism of toxin-like production and of replication. I would to say to everyone my personal experience with SARS-CoV-2. I work in lab and I investigated personally SARS-CoV-2. Okay, so I make the I make the bacterial culture, and I made it all the preparation from the stool sample to the proteomics profile, and I get infected 3 times during this pandemic. Okay, the first time was very heavy. Okay, uh, because the first time I don't know to have COVID, because it was in the November of 1 month before proclaimed the pandemic. So in the November of 2019, I treated in empirical way with azithromycin and rifaximin because I have problem in my gut microbiome. The second and third time, I wouldn't to take first of all the virus because I want to experiment on myself our combination. So like every scientist making the world. So to observe what would be do if I take antibiotics of amoxicillin and rifaximin. The second time, my illness was of only 5 days, and the third time is only of 2 days. But every time I take antibiotics, I have no problem. I have no asthenia or other problems, neurological problems. I feel well. I take SARS-CoV-2 3 times, and we are prepared also a paper about pregnants, because we have treated also pregnants with SARS-CoV-2, and their child are very, very well with the solution of antibiotics, amoxicillin and rifaximin. So it is very important, this mechanism. The toxin can bind into the neurological receptor, nicotinic and muscarinic receptor. I want to say to everyone that you don't have more fear. You don't have fear to contract SARS-CoV-2, because there are the solution. There is the solution. And if you have long COVID, okay, work on your microbiome. Work with rifaximin and probiotics. The secret is reset with antibiotics and restore with probiotics.
Thank you very much. That was very useful, because there are people who are afraid to take antibiotics, and I really wanted to show the complexity of the process which is happening in the gut microbiome. And there are numerous publications now which are studying stool samples and say that virus replicates over a year among patients who suffered SARS-CoV-2. So there is a viral replication going on unless you stop it. And the only way to stop it is with the use of antibiotics. If you experience severe symptoms long after you had SARS-CoV-2 infection, this is an option to consider, try, but please consider restoring your gut microbiome afterwards, because this is very important as well. You need to increase your diversity in microbial species. You need to take probiotics. You need to increase your fiber intake. There are solutions. Speak to your nutritionist. There will be proper ways to restore it and help you recover properly. Carlo, thank you very much for your time. If there is anything else you want to tell to people, so it's your opportunity.
Don't have fear.